Hello, this is Dr. Dan Guerra from VerEvMed. Today we're going to have a new lecture that's going to be discussing some very important breakthroughs in looking at biomarkers and possible pharmacological targets in heart failure. So why don't we just get started? So sometimes I use these images and I want to make sure that I give them good credit. This was actually a picture taken from my son when we were in Utah last summer. And so most of the images I use are from our original portfolio of photographs. And when I don't, of course, I acknowledge where I get them. But I wanted you to know that this was taken by my son. So why don't we go ahead and get started with the actual lecture. So I'll put on my glasses. Okay, we're going to answer, hopefully, a question today, and that is diminished fatty acid oxidation linked to heart failure? So I like to pose it as a question. In most of my lectures, this is what I attempted to do when giving lectures at university. And that's because if we try to focus on our attention to a particular topic, if we pose it as a question and then we give the lecture as a series of answers to that question, it's more affordable in understanding. And I think that that's the important thing about conveying knowledge, is that while we're conveying that information, and so that it can be assimilated as more than information, but is actually the presuppositions to knowledge, that we need to be able to formulate it in such a way that the people that are listening to it see it as something that is trying to seek out something, trying to understand something, rather than just be given information just like a computer would give you. And that's the difference between machine learning and having a human being talk to you, amongst other things, of course. So that's the question we're going to ask. Again, I'm Dr. Dan Guerra, and I'm the Chief Science Officer and co-founder and co-owner of VerEvMed. And who we are scientists verifying published evidence in biomedicine. So that's the question. What's the topic that's going to result in answering that question is going to be more discrete and specific. Specifically, we want to talk about acyl carnitines and heart failure. And so we're going to dig deep into a paper, actually a couple of papers, and in doing so, be able to answer the question that we formulated at the beginning of the lecture by demonstrating that acyl carnitines do say something about heart failure in humans. This, again, is one of our images. Okay, the first paper we're going to look at is the European Journal of Heart Failure. Yes, there is such a journal. You can see this paper came out only about 3 months ago. I'm recording this in October 2017 from VerEvMed Studios, the Pacific Northwest. So let's go through what we know about heart failure, and then we'll get into the paper that we're going to try to elaborate on this morning. So chronic heart failure affects well over 10% of people of 60 years of age or older. So there are different types of heart failure, but chronic heart failure is probably the most common. Heart failure in general is associated with reduced ejection fraction. And you can see there, that's the symbolism for that. And that remains the most common form responding to current surgical and medical therapies. However, there is heart failure associated preserved ejection fraction. So this is different from the reduced ejection fraction, which is more common. There's the acronym for that. It's been less responsive to current evidence-based practice. So we want to know whether or not these two have similarities and differences that can be looked at in terms of clinical therapeutics. We already know that they present differently, right? Because there's difference in ejection fraction. Do they respond differently because of that? And what are the underlying physiological and biochemical correlates that result in having reduced ejection fraction versus preserved? And can those be pharmacological targets? Or indeed, can we find biomarkers to separate the two out and then eventually discern pharmacological targets. That's going to be addressed today. So back to general description, heart failure is a progressive disease. So people that get old, the older you are, if you have the original symptoms of heart failure, it's likely to get worse as you age. Now, we also have acute heart failure, and that involves basically the same etiology and same presentation, only it's rapid onset deterioration in cardiac function. So it's not the kind of thing that you would see in regular checkups year after year. It might be something you see more in the emergency department, right? So what are some of the pathophysiological associations with heart failure? Well, there's congestion. That's probably the most common clinical sign, especially when someone presents with acute heart failure, but it's also something that presents with chronic. And you also have an association with that peripheral hypoperfusion, which means decreased blood flow because of this problem with maybe congestion or with other kinds of comorbidities associated with heart failure. You also get something called cardiogenic shock. So those are the three main clinical signs. You get congestion, peripheral hypoperfusion, and cardiogenic shock. All three of those are common clinical manifestations in heart failure. So when you get congestion and or hypoperfusion, that can lead ultimately to multiple organ failure and indeed death. That's because of the decreased blood flow. It involves a decrease in cardiac output, and one of the things that happens when you have decreased cardiac output is you start to get extravasation of water and whatever's dissolved in that water from the capillary blood vessels. So that results in what we call ascites fluid. And the ascites fluid can then cause swelling or edema, and that's actually another one of the symptoms that are presented when a person has heart failure. That's the last thing on this slide. So the presenting symptoms are otherwise rather general: shortness of breath, even after, like, say, a short walk or semi-strenuous exercise. Muscle fatigue, again, you can see how this is related to blood flow because of lack of oxygen and nutrient supply to the skeletal muscle. And also, as I said, edema because of this extravasation. And that can be really important because of what actually results in getting out of the tissues and into the blood. So here's a paper that I want to focus on mostly today. It's from the American Journal of Physiology, and this is the heart circulation physiology subdivision of that journal. The paper was in press in mid-July this year, and the authorship is Ruiz et al. Most of the researchers on this paper, as I recall, are Canadian. Heart failure, this is now from their paper, rewording the text so that we can present it easier in a lecture form. Heart failure, of course, is a disease where age, sex, and comorbidity must all be considered when examining the underlying disease and respective etiology. So these could be confounders when we're trying to do a cohort study while looking at heart failure, even compared to one type of heart failure patient and another. So you have to be able to know whether or not the age is causing a particular associated pathology. We want to know if sex is associated. Men and women are different in their heart failure presentation and also in outcome. And also, of course, any comorbidities. And those comorbidities are the ones we see most often in the clinic in this year, in the years around this time. We see a lot of obesity. We see a lot of obesity-linked disease and comorbidity. So these include things like cardiovascular disease, metabolic syndrome, hypertension associated with metabolic syndrome, and of course, type 2 diabetes and other kinds of metabolic disorders. So those are all things that have to be taken into consideration, which could be confounders in any good study. So one of the key things as a biochemist that caught my attention about heart failure is that bioenergetics plays a key role where fatty acid beta oxidation, what they call beta ox, impairment is linked to most forms of heart failure. Okay, so we're going to get into a discussion of biofuels, particularly fatty acids as biofuels. Now, right away at onset, I want to make it clear that the heart, because it's constantly pumping and it's constantly contracting, requires constant source of energy in the forms of course, ATP. The easiest way to make ATP the most efficient is aerobically, and that requires, of course, oxidation of NADH to NAD and oxidation of FADH2 to FAD plus, using the electron transport chain in the inner mitochondrial membrane, and via the proton pumping ATPase generating ATP in oxidative phosphorylation, or OXPHOS. So I'm going to, throughout this talk about ETC, there's the electron transport chain, and OXPHOS, which is oxidative phosphorylation. Those are linked, of course, when you oxidize NADH or FADH2. And we're going to find out that the beta oxidation of fatty acids results in the production of NADH and FADH2. So there's your link. Okay. And of course, since oxygen is the terminal electron acceptor, the electron is being driven off of the fatty acids into NAD to make NADH and then ultimately into molecular oxygen, as well as the reducing power associated with that, that a limitation right here that you can see in heart failure is if you get poor flow rate, you're going to have a decreased amount of oxygen to the tissues. Decreased amount of oxygen means there's going to be a limit to the amount of beta oxidation. Okay. So now, right away, then we can talk about fatty acids. Now, in many pathologies, many diseases, not just heart disease, circulating free fatty acids are lipotoxic. And they're lipotoxic for some very general biophysical reasons. The main reason is that fatty acids, free fatty acids, are very detrimental to membrane integrity. So they can disrupt membrane integrity, and once you do that, you'll cause membrane leakage, you'll cause cellular content leakage, you'll also cause a perturbation of what is expressed or supplied on the surface of cells, and they can become immunogenic to the immune system. So anytime you get free fatty acids in any reasonable concentration, because they're not water soluble, they will associate with a membrane fraction, which is, of course, lipid soluble, hydrophobic. And when they do that, the free fatty acids can disrupt the membrane integrity. So because of that, the cell intracellularly captures most of the fatty acid that's inside the cell and makes one of two kinds of ester. A thioester with coenzyme A, and these are called acyl-CoAs, or oxygen esters with carnitine. And carnitine, as we will see, is a derivative of trimethyl lysine, okay, from amino acid metabolism. Carnitine plays a very special role in the later stages of beta oxidation, as we will see soon. So once you make acyl-CoAs and acyl-carnitines, you are able to target the fatty acids for beta oxidation, therefore for bioenergetics, therefore for heart contraction, muscle contraction. Okay. So if you are limiting the amount of fatty acyl-CoA and fatty acyl-carnitine to the cardiac muscle, that could limit the amount of energy that heart muscle is going to be able to derive, right? So that's known as the energy starvation hypothesis for heart failure. It's especially linked then to decreased blood flow, because that's going to limit molecular oxygen, which is the ultimate electron acceptor, as I just said, for the flow of electrons from the fatty acids, ultimately to oxygen, and then ultimately the proton pumping ATPase to make ATP for oxidative phosphorylation. Curd paper then is going to look at fatty acid energy substrate paradigm. Okay, so that's what they're interested in looking at here. And I want to compliment the authors right away and say it's a very elegant work, very detailed. They used modern technique of liquid chromatography, tandem mass spec, to be able to isolate metabolites. So basically, this is a metabolomic paper. Okay. So first thing I want to do is just clear the deck and make sure you understand what beta oxidation is. There's no sense in me going on and telling you what their data looks like unless you look at what I mean by beta oxidation. So I'm a biochemist and I love looking at pathways, and particularly with structures in the pathways. So if it's something that turns you off, you try to see why it's really useful to understand this, because then you get a complete three-dimensional view, and if it's through time, four-dimensional understanding of what we mean when we talk about metabolic pathways and how they are linked to basically all disease states and healthy states for that matter. So this diagram here shows you that we've already made a thioester. We made a CoA thioester. Now that activates this fatty acid, puts it in a metabolic play. That's a very active center there, that thioester, that carbonyl associated with that sulfur atom. Okay, we call that an acyl configuration. Basically, it's a radical configuration. So what happens, the first thing in beta oxidation is we're going to oxidize the fatty acid. And when you oxidize something, you reduce something else. Now you're going to reduce this flavin adenine dinucleotide, okay? And you're going to make FADH2. And that enzyme, acyl-CoA dehydrogenase, that FADH2 that's made is going to go off the electron transport chain, as I just mentioned. And when it does that, it's going to get oxidized. The electrons are going to move through the electron transport chain through various ubiquinol pathways and cytochromes and heme-containing proteins. And ultimately, in that intermitochondrial membrane, those electrons are going to be driven along with the proton pumping force of the electron motive force of protons moving out of the mitochondria and back in from the intermitochondrial space to generate ATP via the ATP synthase, the proton pumping ATP synthase, which is actually complex 5 in the electron transport chain oxidative phosphorylation pathway. FADH2 will only give us 2 ATP, but it's still plenty, and you already are there with just one reaction. The next reaction is called a hydratase, and we're going to pass water over that trans double bond. That's what we're going to hydrate it. And you see here what that ends up giving you is a hydroxy fatty acid. There's no H group there, right? So that's an alcoholic functional group there. So what happens after that, now that can build up. By the way, hydroxy fatty acids can build up. And when they do build up, they can result in toxicity. And then we're going to see that, okay? But I just want to point out that that's one place where you get hydroxy fatty acids. So key point here is that this hydroxy fatty acid, when you're going through the beta oxidation pathway, you're going to go through another dehydrogenase. Only here there's enough energy in this system here, okay, to be able to drive NAD reduction to make NADH. The NADH goes again through the same exact electron transport chain. It starts earlier, complex 1 actually. And when it does so, it's able to generate 3 ATP by the transport of electrons through the ETC and by the proton pumping, ultimately through OXPHOS to make ATP. Okay, so you've already now made 2 plus 3, 5 ATP, and you're not even finished with the beta oxidation pathway. Anyway, what you end up now is a completely oxidized keto, that's a keto function, acyl-CoA, okay? And ultimately then you're going to get a thiolase reaction. This thioester is going to come in, it's going to cleave off those first 2 carbon atoms. This is the beta elimination, because remember, look back up here, this is the alpha carbon, that's the beta carbon, okay? So that it's called a beta oxidation, because from that beta carbon, these 2 carbons are going to be released as acetyl-CoA. There they are. So now you're left with a fatty acid 2 carbons shorter, and you're ready to do another round of beta oxidation. I've talked about this in other lectures for VerEvMed, and of course, over the last 30 years, probably thousands of times in biochem lecture to graduate students and medical students and whatnot. But at any rate, this acetyl-CoA then can either go through the tricarboxylic acid / Krebs cycle, make more NADH when it condenses with OAA to make citrate, and then through the dehydrogenases, which are embedded in the TCA cycle, or they can generate ketone bodies. Ketone bodies, and we'll show you ketogenesis in a moment. That's when you're taking the carbon that you've removed from the fatty acid and you're making a soluble hydrocarbon that can enter the bloodstream and be used by all the peripheral tissues, and including the brain, including the heart. And in so doing, you are able to oxidize that carbon one more time, and that oxidation will be through the TCA cycle, and you'll make a lot more NADH and FADH2, and then therefore more ATP. So ketogenesis is a very important aspect of this. So this is the beta oxidation of fatty acids, and of course it drives the energy currency or energy budget of the cell. Okay, so you have other kinds of oxidation of other kinds of foodstuffs, of course, like glucose. Glucose doesn't require molecular oxygen for glycolysis to oxidize it, but the yield of ATP, because you're not using oxygen as the ultimate electron acceptor, is way lower, way less efficient. So fatty acid oxidation, the reason, if you want to call it a reason, the purposing of it for heart contraction is that it's a steady, constant, high yield flow of ATP when you oxidize fatty acids, and as long as you have molecular oxygen. And a healthy heart, there's plenty of molecular oxygen because there's plenty of oxygen dissolved in the blood. That's what the heart's doing, right? It's pumping that blood around, drawing up all the oxygenated hemoglobin from the lung and pushing that oxygenated hemoglobin to all the periphery of the body, all the peripheral tissues of the body. So there's going to be plenty of oxygen. As long as you have plenty of oxygen, you have fatty acid. The fatty acids are going through their esterifications, and therefore they are supple reactants to beta oxidation, then there's going to be a much higher yield of ATP that way. And that's what happens in the heart. Okay. So that's all going on in the mitochondrion. There's another really important organelle, which I've talked about in previous VerEvMed lectures, one that is has a sweet place in the heart of all of us lipid biochemists, because the peroxisome is a place where a lot of interesting more mysterious fatty acid metabolism occurs. When I say mysterious, not so mysterious. It's just that it's not often derived in normal discussions in biology classes because peroxisomes, again, you don't find these in prokaryotic organisms. They're only in eukaryotes, and they play a very significant role in animal biology, and particularly in human healthy physiology. So peroxisomes are unique organelles. They're essential in the eukaryotic system, and they're responsible for multiple biochemical pathways, either in conjunction with the endoplasmic reticulum or mitochondrion, or both, or as a separate and independent enterprise. So peroxisomes are really important organelles, and probably a lot of people don't know much about them. We're going to learn a little bit more about them today. I have talked about peroxisomes in previous lectures, so you can go back and look at those. And when I finally get around to cataloging them on the VerEvMed website, then you'll be able to then pick those out, because I'm going to set that up soon. I've got somebody working with me on that. So the relevance of peroxisomal activity in the cardiomyocyte, which is germane to the discussion today, is best understood for its vital role in fatty acid and complex lipid metabolism. And that's what we're going to find where it's an important element in the paper we're going to look at today by Ruiz et al. By the way, I first heard about this paper on a podcast. So I listened to a lot of medical podcasts and scientific podcasts. I heard the authors actually being interviewed on the paper. I said to myself, gee, this is really a good paper to put out for VerEvMed. So that's where I got the idea from. Plus, as I said, this is a client-based discussion because we have a client who has interest in knowing about heart failure. So the peroxisome, and in fact the endoplasmic reticulum, do something different than beta oxidation. They also do that, by the way, but they do omega oxidation. So in order for you to understand that, you know that these are Greek letters, right? And omega is the last letter of the Greek alphabet. And so that means that the last carbon on the fatty acid is where we're going to get this oxidation. So it's kind of unusual, right? And the last carbon on a fatty acid, on a long chain alkane like this, monocarboxylic acid, is going to be a methyl group. So you're going to have a mixed function oxidase, which is going to pick up molecular oxygen here. We're going to incorporate it into that methyl terminal omega carbon, and we're going to make a hydroxy fatty acid. See, that's a fatty omega, because the last carbon, because remember the first carbon is that carboxylic acid, then the alpha carbon is somewhat paradoxically called the alpha carbon, the second carbon in. Well, the last carbon is called the omega, last one, right? All right, at any rate, Now you've made a fatty omega hydroxy acid. Now this is going to be an important player in today's paper. Okay. But you don't stop there. You can continue on with the oxidation process. Now watch what happens in the peroxisome. You have this fatty omega hydroxy acid. Then you react it with two enzymes, which are very common, alcohol dehydrogenase and aldehyde dehydrogenase, same ones that actually work on ethanol, right? So here you would have ethanol going to acetaldehyde and acetaldehyde going to acetone, actually, or acetic acid, depending. But here we're going to be taking this preformed fatty acid, which is the hydroxylated. So you got a fatty omega hydroxy fatty acid. Now look here, we make NADH, right? So we're going to oxidize. We've oxidized this now to what is that? That's an aldehyde. So this is a fatty omega aldo acid. There's the acid portion. Okay. And we've made NADH. Now that reducing power can be used for ATP synthesis, not in the peroxisome, but the reducing equivalents can be shuttled, the reducing equivalents, not NADH, as a molecular entity, can be shuttled to the mitochondria and ultimately used for, oxidative phosphorylation. So, and plus NADH is really critical inside the peroxisome for dealing with redox potential in that peroxisome. Because the peroxisome's named because it has a lot of peroxide in it, and that means that there are a tremendous amount of free radicals that can be generated in the peroxisome. You need a lot of reducing equivalents. NADPH plays that role usually in the cytosol, and NADH can play that role in the peroxisome. Okay. Now, carrying on, you have the aldo acid, and finally you get the second oxidation. It's an aldehyde dehydrogenase. Again, you have enough energy to be able to reduce NAD to NADH. So you can make 2 moles of NADH here. And finally, you end up with a fatty dicarboxylic acid. This is going to be a really important player in today's talk. So you've made you've made an omega fatty acid, okay, omega hydroxy fatty acid, and you've made a fatty dicarboxylic acid. These two species are going to make acyl carnitines, and they're going to be important in this Ruiz et al. Paper, 2017 paper. This is just the same pathway again, and just so you understand, dicarboxylic acid so formed, there they are. There's a carboxylic acid there, and there's a carboxylic acid there. That's at the first carbon, that's at the omega carbon. Right? They can actually undergo beta oxidation now, and they can be reduced down to a C6 compound called adipic acid, or all the way down to succinic acid, C4. Okay. So much to understand, these can be further metabolized, and more NADH and FADH2 can be made via traditional or conventional beta oxidation. So the peroxisomal oxidation of very long chain fatty acids is what is really critical here. So the peroxisome is a key organelle associated with the plasma lemma, or the plasma membrane, the outer membrane of the cell. Okay, so the outer membrane of the cell has a lot of unusual molecular species of lipids. Besides the phospholipids, which are in the glycerol family, you also have sphingolipids. Now, those lipids in the plasma membrane sometimes have very long chain C20, C22 fatty acids. And when you find those very long chains, sometimes polyunsaturated fatty acids, those are very important in bioactivity of that membrane. That is, what the membrane's supposed to be doing requires those very long chain fatty acids. Now, many of those long chain fatty acids are inundated with multiple double bonds. So they're very long chain polyunsaturated fatty acids, or VLC PUFAs. Because they have a lot of double bonds, they're prone to autooxidation in situ within the membrane. If they're autooxidized in the membrane, you generate radicals such as hydroxyl anions, such as superoxide, such as hydrogen peroxide. All of those are bad players, bad agents in the membrane. Likewise, the oxidized fatty acid no longer functions correctly. So that causes membrane disruption. Now, because of that, you have to turn over those VLC PUFAs regularly, and that's one of the main functions of the peroxisome, to pull down those fatty acids that are becoming oxidized from the plasma membrane, and then rebuild those very long chain polyunsaturated fatty acids. Okay, so fatty acid synthesis now to be able to rebuild and plug them back into the membrane. So you have to, like, in other words, you have to remove older, very long chain polyunsaturated fatty acids and replace them with newly synthesized so that you reduce the amount of oxidized fatty acid in the membrane. That's one of the jobs of the peroxisome. Okay, so just so you understand that. In peroxisomes, then we see that there's a flavoprotein dehydrogenase, and that's going to transfer electrons to oxygen. And what it's going to yield, okay, right away, this is going to be a different kind of dehydrogenase. You're going to make hydrogen peroxide. So you have to get rid of that hydrogen peroxide, okay? The way that happens, in fact, that's why this is called the peroxisome. It got its name because a lot of hydrogen peroxide staining was found when the first people were studying this organelle. Easy to stain for hydrogen peroxide, by the way. There's an enzyme in the peroxisome called catalase, constitutive enzyme, and that just breaks down hydrogen peroxide, and it makes water and oxygen from hydrogen peroxide. Okay, so that's because hydrogen peroxide can be toxic. So you might ask, why does the cell do this? Well, actually, hydrogen peroxide toxicity is also important as one of the components that epithelial cells do, and indeed immune cells do, when their peroxisomes are actively involved in bactericidal activity. So just like you use hydrogen peroxide in an open cut, the cell generates hydrogen peroxide, and in one of the aspects of the innate immune response, that hydrogen peroxide is used to kill intracellular bacteria. Okay, so it's one of the reasons that H2O2 is made. But it's not good that you build up H2O2, so there's a very active catalase, right? All right. So subsequent steps are identical with the mitochondrial counterparts. I already mentioned that, and they're carried out. The oxidation, as the regular beta oxidation, carried out by just a different isoform of those enzymes, the ones that end up in the peroxisome rather than the mitochondria. So here again, just showing you is the peroxisomal oxidation of very long chain fatty acids. Here you're talking C18, C20, C22, even C24 fatty acids. So there you go. There's your trans double bond. Here's your acyl-CoA dehydrogenase here. Okay. There's your trans double bond there. Here's your saturated fatty acid, right? So this is just looking at your conventional. This isn't an omega oxidation. This is your typical beta oxidation type of system, right? This alpha carbon, beta carbon, right? And there is your carboxylic acid group, the very first carbon. At any rate, acyl-CoA dehydrogenase is going to make H2O2. Catalase is going to remove that toxicity, as I just mentioned to you. And then you're going to make this trans fatty acid. It's going to go through further oxidation. So the specificity of the peroxisomal enzymes is for longer chain fatty acids. If you take an extract from peroxisomes and you try to give them shorter chain fatty acids, they're very poor substrates. So again, it describes the detail of what the peroxisome's function is, to deal with removal of very long chain polyunsaturated fatty acids. And that's exactly what its substrate specificity is. Yeah. All right. Finally, we can talk about carnitine. So once you have an acyl-CoA and you want to target it to the mitochondrion so that you can make NADH and ultimately ATP, you have to first of all get that fatty acid esterified to coenzyme A, as I mentioned. The fatty acyl-CoA then reacts with an enzyme called carnitine palmitoyl transferase 1, which is in the outer mitochondrial membrane. That's going to do just what the name says. It's going to transfer the acyl group to carnitine. So carnitine is then going to be used to synthesize acyl carnitine. That acyl, again, is just that active radical fatty acid, that carbonyl fatty acid, from a thioester to an oxygen ester. Okay, very soluble compound now in that intermitochondrial space. And there's a reason why you don't use CoA esters throughout, because CoA esters, when they reach a high enough concentration, that concentration is known as the critical micellar concentration, or CMC. When you reach a critical micellar concentration, you make a micelle, and a micelle is a unit, kind of a unit membrane closed circle of amphipathic nature, where you have the fatty acids interior and you have the outer portion of it being the aqueous part of the coenzyme A molecule, okay, the phosphopentathene group, basically. And when that occurs, you make micelles, and micelles can also be toxic to cells, okay? So my cells can be toxic to cells. So you don't want to have acyl-CoAs build up to a high enough concentration to make my cells. So it's one of the reasons that coenzyme A itself, and plus it doesn't traverse the membrane, because it has a strong detergent effect. So you make this acyl carnitine. By the way, this is inhibited by mal-CoA. That's interesting because mal-CoA is the first product of de novo fatty acid synthesis. And you don't want to have the constant beta oxidation, which is where we're going here during fatty acid synthesis. So it's a back channel feedback regulation using the first product targeting fatty acid synthase to now initiate a blockage of beta oxidation. So it's just a subtle, again, just showing you how everything's interconnected in biochemistry. The acyl carnitine then is translocated through the inner mitochondrial membrane. By the way, that's where all the electron transport chain stuff is also embedded. And the second protein also embedded now on the inner leaflet of the inner mitochondrial membrane. So the inner leaflet, we call this the mitosol, the interior of the mitochondria, right? There's a second isoform of CPT, carnitine palmitoyl transferase, is CPT2. It's going to pick up now this acyl carnitine, transfer that back to the thioester, make acyl-CoA. So you take acyl-CoA from the outside, make acyl carnitine intramembranously. You now synthesize acyl-CoA back in the mitosol, and you're ready for beta oxidation. You already saw that pathway. So this is where acyl carnitines are important. Okay. So we also showed you in beta oxidation to make ketone bodies, right? What are the ketone bodies? Are shown here: acetoacetic acid and beta-hydroxybutyric acid. You can see this is an alcohol. You can see that this is a ketone, right? And in fact, these are linked. The synthesis of these are linked via an NADH redox couple, okay? But both of these are very water soluble, make it directly into the bloodstream, and these are excellent carbon sources to be picked up by any cell, including the brain, including the heart, including the muscle, and be readily incorporated into the TCA cycle, ultimately oxidized to make NADH and FADH2, and then therefore make ATP. So you don't lose anything when you beta oxidize fatty acids. Now, the key point here, though, is you're not going to make carbohydrate from fatty acids. That doesn't happen in animals. It happens in plants and in microbes. That's called the glyoxylate pathway. Very interesting pathway. I talked about it a little bit in text form on my Facebook page, and we're going to talk about glyoxylate pathway sometime in the VerEvMed lecture, because I want you to get an idea what that C2 metabolism looks like. Very interesting stuff. But in animals, you don't do that. You don't take the acetyl-CoA from beta oxidation and use it to make oxaloacetic acid and then on to gluconeogenesis, right? You take that acetyl-CoA, and if it doesn't get just transmigrated through the TCA cycle to burn it up to make NADH to make ATP, then the excess of that acetate is going to make these ketone bodies through ketogenesis. Here's ketogenesis. Again, this is happening going down in the mitochondria. The similar reactions occurring in the cytosol with acetyl-CoA, and that's a totally different pathway, and that's the synthesis of prenolipids and ultimately cholesterol. Very interesting how these intermediates play multiple roles. But how the acetate gets out, acetyl-CoA gets out into the cytosol from the mitochondria it has to go through the TCA cycle. You have to make citrate, and you have to run citrate lyase reactions to be able to pull it out. But that's a totally different story. Let's look at ketogenesis. Acetyl-CoA, okay, you got 2 moles of that. You remove one of the CoAs in the ketothiolase reaction, you make acetoacetyl-CoA. Then you add a third acetyl-CoA, so 6-carbon compound here. Sorry for that. Make a 6-carbon compound here called 3-hydroxy 3-methyl-gluteryl-CoA, or HMG-CoA. That's the same intermediate, by the way, the same intermediate in cholesterologenesis, only the intermediate has to be made in the cytosol to make cholesterol. This is in the mitochondria, okay? Particularly in the liver. All right. Now, this HMG-CoA is then lyased to remove an acetyl-CoA. And what you end up with now is the C4, first ketone body, acetoacetate. See? This acetyl-CoA goes back to replug into the system. So you're using acetyl-CoA to make acetoacetate. Notice no more thioester, no oxygen ester. Don't need it. It's freely soluble compound. It's not going to make any micelles at some critical concentration. It's not going to cause any other perturbations in metabolism. It's just a perfect carbon source. It can go right into the bloodstream and act as a source of energy. Okay. So acetoacetate then can spontaneously form acetone, and you get this acetone actually on the breath of people that have a lot of ketogenesis. So people that are going through a lot of ketogenesis, you'll smell acetone on their breath. In fact, alcoholics tend to have a lot of that because alcoholics make a lot of acetyl-CoA. So that's one sidebar to this discussion, but not important today. What we want to know is that if you take acetoacetate and you reduce it, you make the beta-hydroxybutyrate. These are the two ketone bodies I just showed you, right? And there's this couple here between NADH and NAD, between acetoacetate and beta-hydroxybutyrate, and that couple actually describes a redox potential in the mitochondria, okay, because of that NADH. All right, so putting it all together. Fatty acids are indeed amino acids that are ketogenic, will make acetyl-CoA. Here is ketogenesis, right, making the acetoacetate beta-hydroxybutyrate. They leave hepatic mitochondria. They, in fact, leave the hepatocyte, leave the liver, hit the bloodstream. They make it into other tissues, peripheral tissues like skeletal muscle, heart muscle, brain, okay? And these two ketone bodies then, beta-hydroxybutyrate, right? It's going to make some NAD. It's because it's got to be what? It's got to be oxidized, right? And the acetoacetate is going to go through basically a series of reactions that look almost in reverse, although you have to use succinyl-CoA to supply the CoA here. So there's a thiophorase reaction, which makes acetoacetyl-CoA. You've now activated that carbonyl. You do another ketothiolase. You add some more CoA to this. You make two acetyl-CoAs, TCA cycle. Boom, boom, boom. NADH, FADH, 2 ATP synthesis. So that's why these are really useful fuels. All right. Now back to the paper that's most critical for today's discussion. So we've gone through beta oxidation, omega oxidation, ketogenesis, carnitine metabolism, just to recap. Now let's put these things together. So what they did in this paper is they want to do metabolomics. There's two different kinds of metabolomics. There's targeted and non-targeted. And what do you think that means? Targeted means you want to look at something very specific. You want to shine the light on a particular pathway or a particular set of metabolites. Non-targeted is let's throw everything up on the wall. Look at all the metabolites in our system and say, what do they tell us? Can we make ratios out of them, like substrate product ratios, and understand something about metabolic flux, redox, for example? Can we find a whole embedded metabolic pathway when we look at a non-targeted metabolome? Or can we find some unique pathway we didn't know about before, or unique at least to the tissue or the system or the pathology of the system we're looking at? So that's why you might do non-targeted. Targeted, though, gives you we already know a lot of what's going on, so we want to look at something very specific. The folks in this paper kind of did both. They did a lot of metabolites, but the metabolites are all sort of just general metabolic pathway stuff, amino acids, a little bit of carbohydrates, but mostly lipids, fatty acids, right? TCA cycle intermediates. Okay, so basically that's what I want to say, is that there's different ways of doing metabolomics. Metabolomics, though, what it's about is looking at metabolites in a cell, rather than looking at the genome, which is asking what's going on with the genome in a cell, the transcriptome, what messenger RNA is being transcribed, expressed that is, or the proteome, what proteins are made from those transcripts, or indeed specific metabolomes like the lipidome, right? Only looking at lipids. A metabolome would be kind of like the generic parent to a subset of metabolomes such as the lipidome, right? Or the sphingolipidome, right? So that's where you get all of these different nomenclature discussions of OMEs, right? So in general, all of the things, genome, transcriptome, proteome, metabolome, we call that omics, O-M-I-X or O-M-I-C-S, right? We call it omics because it's simpler to write in manuscripts. And basically that describes all of a descriptive way to continue in understanding what's going on in the cell, okay? The metabolomics gets down to the grainy, looking at the granular structure of the cell, what's actually there in terms of organic molecules, right? So that, and you get lots of things you can look at there. So what they're going to look at are the metabolomics. Now, when I was reading this literature, okay, so you're reading a lot of papers when you put together a lecture, not just the one you're looking at, I came upon something I didn't know. So, I am the first person to say, as a biochemist who's been a PhD biochemist for over 30 years now, that I often encounter things when I start digging into literature that I didn't know, right? And I love it because that's what really makes being biochemist stimulating, particularly intellectually stimulating, because you learn things you didn't know, and it helps you incorporate that into a larger knowledge base. So there is something called femininization in metabolism. And I already heard about this because I know that as men age, male mammals age, they tend to lose testosterone. That is the major androgen in men, right? And so that's called a femininization. It's common in endocrine literature, for example. Well, I didn't know that there was something very specific in, heart failure, but there is. So in metabolic heart failure, male patients, okay, there's this feminization. You get this decrease in androgen. Now that's going to have a large pleiotropic, that means a complex, organized, orchestral effect on, that's what I mean by pleiotropic, effect on lots of metabolic pathways. Because remember, testosterone and androgens are hormones, and hormones control the floodgates of a lot of upstream processing of later metabolic sequelae. Right? So whenever you see a hormone change, you're going to get a lot of downstream things that are going to change because of it. That's why people get excited when they hear about it. At any rate, this feminization presents with general cardiomyopathy in men that have this decrease. Not all men do show a twofold decrease. Some men show very little. Some show it only very late in life. Some never show it. Yeah. At any rate, this general cardiomyopathy and the heart failure linked increase in ketone body utilization. Here you go, ketone body. So that means a lot of beta oxidation is going on. Maybe that's an unusual event, because why would you have all this ketone body generation and not a lot of just NADH production, particularly when you have heart failure, right? So this is a key feature of this, right? All right, so that's kind of a sidebar, but it's interesting. I learned that, and I thought I would turn you guys onto that. So metabolomics has also revealed, so this is just them talking, these people in this paper talking from their introduction, that the heart failure that has the reduced ejection fraction and the preserved ejection fraction. Remember, those are the two different heart failures, two major differences in chronic heart failure. Both of them present with an increase in long-chain acylcarnitines. And that suggests a possible significant role, because it's long-chain and it's acyl groups, carnitines being made for peroxisome malfunction. So that's a unique feature of this paper, and that's why I like it. It talks about mitochondria and it talks about peroxisomes. Okay? So good subcellular cell physiology, cellular pathophysiology. Again, the authors should be commended. They did a great job here. So what do? They determine circulating acyl. So back up. The literature is replete with a discussion of inborn errors of metabolism that deal with high levels of acylcarnitines that build up in hereditary mitochondrial fatty acid oxidation defects. Okay, so there are inborn errors of metabolism, otherwise known as genetic defects, and in people, young children who suffer from problems with having an intact beta oxidation pathway, they build up what? The precursors to the oxidation, and one of them would be acylcarnitines. Okay, and in fact, because you have thioester, oxygen ester, the way that the reaction dynamics work is that the oxygen ester is going to be favored in that mediated transfer of the acyl group from the CoA to the carnitine. And again, maybe one of the reasons the cell does it, teleologically talking, is because if you build up enough CoA anywhere, including inside the mitochondria, you're going to, CoA esters that is, you're going to get this critical micellar concentration breached, and then you can cause membrane disruption, and of course that's going to cause the cell to die, right? Or maybe make mutations. So it's not good. So acylcarnitines don't do that. So they're a good reservoir for acyl groups that otherwise aren't being oxidized. And that's why you see them in these hereditary diseases. So that's already been determined, and we know that. And these authors are saying, well, because of that, we know that we can determine them. We can get them in the metabolomic profile. So intracellular acylcarnitine accumulation may help decrease the detergent effects of micellar acyl-CoA. This is me talking here, while increasing mitosolic beta oxidation substrate availability. Okay, so if you got acylcarnitines, they're going to be ready to go, right? And you're not going to be building up acyl-CoAs outside the mitochondria or in the intermitochondrial space, for example. Acylcarnitines, however, have another unique chemical aspect. They can be translocated to the bloodstream. Okay, and therefore, because there's actually a protein, we'll see in a minute, that will allow the acylcarnitines to leave the cell and indeed to be taken up by cells. That doesn't happen with acyl-CoAs. Acyl-CoAs do not migrate. They don't move around within membranes because they can't make it through membranes because of the detergent effect. All right. So anyways, they can make it to the bloodstream, these acylcarnitines, and they can potentially become part of what I call a fatty acid bioenergetic homeostasis. So it's my term for this. So if you got acylcarnitines, those are like floating rafts of potential utilizable carbon. Now, they're not ketone bodies, they're acylcarnitines, but the acylcarnitines are maybe even more poised to go into beta, they're ready for beta oxidation. So the ketone bodies are done with beta oxidation. They're ready to just hammer into the TCA cycle, make NADH and FADH2 and make ATP. That's definitely useful fuel. But if you're going to build up something that's during fatty acid oxidation because you have a deficiency or you have a lack of proficient beta oxidation, maybe because of deficiency in oxygen, such as in heart failure, then acylcarnitines are a good thing to kick out into the bloodstream so they can be taken up by peripheral tissues and utilized. So that, again, is a teleological perspective. So I call that fatty acid bioenergetic homeostasis, for lack of a better phrase. So while mitochondrial beta oxidation is directly linked to oxidative phosphorylation, peroxisomal, very long chain, or dicarboxylic acid, or indeed omega hydroxy, fatty acid oxidation may serve complex roles in membrane and signaling lipid metabolism. So what I'm not talking about today, right? Thank God, maybe in a way, because it'd be way longer talk, is the peroxisome, while it's doing all this really cool stuff with removing damaged oxidized fatty acids from the plasma membrane, it's carrying out dicarboxylic acid fatty acid synthesis and omega hydroxy fatty acid synthesis for a totally other purposes. It's purposing those to actually make other metabolic pathways turn on. One of them is actually complex lipid metabolism. Okay? So I want you to understand that those intermediates are used for totally different associated tangential pathways, but not just simply bioenergetics. Okay? Cell never wastes its time with just one major purpose. It has multiple purposes for everything it does. So this paper looks at the contribution of alternate fatty acid oxidation, that is peroxisomal, and subcellular zonation, that is looking at mitochondria or peroxisome. It's called subcellular zonation, okay, as potential loci, that is specific places to look for metabolic fuel dysregulation in heart failure. Here are their methods. They drew blood samples from humans. They're collected in an EDTA tube. Don't worry about that. It's just basically to keep calcium chelated so you don't get the blood to denature, basically. And they put them down at minus 80, right? So that you don't have any biological activity down there. Primary cohort of humans were from the Montreal Heart Institute. They had two basic groups. They had a control, and then they had 68 symptomatic heart failure patients. They used liquid chromatography mass spec, that's tandem mass spec. This is your way of doing metabolomics these days. Sometime I'm going to give you an entire lecture just on how LC mass spec works. It's really a cool thing, especially for people that have engineering background or physics background. You're going to really like the discussion of how the mass spec works. Not going to do that today. Anyways, it's a typical instrumental analysis. It's a lot of people can do this. A lot of laboratories can do. You get benchtop now, LC mass spec. It used to be a mass spec would take up a whole laboratory room. When I was a postdoc in Peoria, Illinois, working for the Agricultural Research Service, the mass spec there took up an entire room, probably like 2,000 square feet of space, right? Nowadays, a mass spec, a tandem mass spec, can basically sit on the benchtop in a lab. Okay. So technology at work. Okay, so they looked at a lot of things in terms of metabolomics. They wanted to look at lactate to pyruvate ratios and beta-hydroxybutyrate to acetoacid. And you already saw that. That's going to give you an NADH and a decouple. So is lactate to pyruvate. This is going to give you redox in the cytosol because of the enzyme there is lactate dehydrogenase. And there's a lot of other reasons to purpose lactate to pyruvate, because lactate leaves the cell and they can migrate and it can be reconverted to pyruvate. Pyruvate can then be driven through the pyruvate dehydrogenase reaction, oxidized to acetyl-CoA, decarboxylating, and then acetyl-CoA can have a TCA cycle. Sorry, that's way too much information there. But anyways, that's cytosolic redox state, crudely, I would have to argue. And then beta-hydroxybutyrate acid is also rather a crude mitochondrial redox state, but it's okay. Anyway, I will just tell you, kind of like a killer alert on you're going to learn something already about the data. They didn't really find anything really too sophisticated about these ratios. They didn't see any difference between the control group and the symptomatic HF people. So, all right. Anyway, they did look at acylcarnitines, major focus in the paper. They looked at acetylcarnitine, which is just acetate, C2. Short chains, which you're calling C3, C4, C5. See, there are some chain. Medium chain are all even, 6, 8, 10, and 12. Long chain, they're calling 14, 16, 18. Fatty acid biochemists like myself wouldn't call 14 a long chain. We'd call 18, 20, 22, 24 a long chain, but for purposes of what they're describing here, it's okay. Then they also looked at those hydroxylated acylcarnitines. Remember, the AC is acylcarnitine, and they looked at decarboxylic acid acylcarnitines. Again, those are peroxisomal products, right? Here's the data. Basic thing here on the y-axis, you're looking at the ratio of deuterated internal standard. So one of the ways you do this is you have an internal standard. They're using deuterium, which is a heavy isotope of hydrogen. It's a really good way to do mass spectroscopy because remember, you're looking at mass, and deuterium is heavier than the proton, than hydrogen itself, right? It's a heavy isotope. So that's a good way to use as an internal standard to look at that isotope, heavy isotope migration, without going into detail again about how mass spec works. So basically what I wanted to show you here's all the different species of acylcarnitines in the blood, remember we're looking at. And wow, look at this. So the more asterisks you have, the more significant it is. You get a lot of carnitine or a lot of C2 acylcarnitine. And indeed, all these species, when you blow up this mumbled data here and you increase this y-axis, you see there's significant increases in acylcarnitines at the C4, C5, C6, C10, and C14 carbon level, with heart failure having higher amounts here on these long short chain ones, and then the very long chain, very significant. Okay, that's again a key feature here. Again, why are they talking about that? Peroxisomal activity, right? So it looks like indeed heart failure patients have a lot more acylcarnitines in the blood of all species, and it's significantly different. That's what you're looking at. I don't like these standard error bars, although I like to see them. It means there's a lot of standard error, but I think that is sort of endemic when you look at blood. When you look at a crude liquid biopsy like that, you're going to get a lot of variation, right? But it's how it goes. When you're trying to get a really simple biomarker, sometimes you have to deal with a lot of variation. So, but it's significant, so they had enough numbers. All right. Now, remember I told you about the confounders? I said that there's a lot of comorbidities in heart failure. One of them is type 2 diabetes because that's associated with obesity, and a lot of obese people are associated with that also have heart failure. Well, it doesn't look like type 2 diabetes explains the variation in the acylcarnitines, which is basically what this data says. Look at that. No significance except at the acetylcarnitine. And maybe that's because you just have a whole lot of acetate around. And so maybe that isn't significant. Maybe it is. It's worth following up. But you don't have anything in those medium chain acylcarnitines, and you have nothing in the peroxisomal activity. So diabetics or non-diabetic heart failure patients, no difference. So that confounder can be bracketed out. It doesn't look like whether or not you have diabetes is this acylcarnitine going to make a difference. Now, why is that significant? Because you see, if you're trying to key out, you're trying to isolate just heart failure, right? So it's not that these people, many of them, I forget what the percent was, a lot of these people had diabetes, these HF patients did. So that's kind of a common comorbidity. But you want to look at heart failure specifically, right? So you don't want to just be looking at what diabetic blood looks like in terms of acylcarnitine. You want to be able to link it specifically operatively to heart failure. That's why they do this kind of work, right? Get rid of those confounders and say, this looks like it's heart failure associated more than diabetic associated. And that's what you see. They want to emphasize the biggest differences is, again, their data. I'm just running through their data. The biggest differences are what they call long chain acylcarnitines. Okay? Now that's important because, sorry again for that. That's important because it means what? Peroxisomal activity. Okay? That's what we're saying here. Because these are all being derived from peroxisomal oxidation, and presumably that's why you see these very long chain acylcarnitines, right? They're coming from peroxisomal metabolism. All right. So there are also significant increases, again, it's peroxisomal of hydroxy and dicarboxylic acid acylcarnitines. Again, peroxisome, long chain fatty acid, omega hydroxy fatty acid, dicarboxyl fatty acylcarnitines, right? All three of those molecular species of acylcarnitine look like they're peroxisome associated. And that's what you get. Big differences. See this big, here's the heart failure people. Heart failure people are the gray columns, and the white columns, the clear columns, are the control patients. Significant here at C8, C10, these are hydroxys. At the C6 dicarboxylic acid, and indeed at C16 and C18 dicarboxylic acids, whopping more acylcarnitines than you do in the controls. Okay, so it looks like this is a heart failure associated peroxisomal dysregulation of fatty acid metabolism, basically what they're saying. So this allowed them to generate this model. That's how their paper ends. You get long chain fatty acids entering the mitochondrion, okay? They're esterified to coenzyme A, right? They're picked up with carnitine. They go through the carnitine pathway. This is all beta oxidation. Because there's a lot of carnitine, you make a lot of long chain acylcarnitines, okay? So there's a sufficient amount of carnitine. We're going to see why that is. I don't think they address it in this paper, but I'll explain it to you. But you get a lot of carnitine. The high level of carnitine means that you can, especially if you're getting a decrease in beta oxidation, I told you that you're going to get a buildup of the precursors for the pathway. You're not likely to get a buildup of acyl-CoAs because they're toxic. You're going to get a buildup of acylcarnitines, and they can actually leave the cell, right? Leave the mitochondria, leave the cell, and leave the whole organ, right? And that's what they found, remember, because it's in the blood. They're finding it. So they're just saying that when in the heart failure patients, you get a lot of long-chain acylcarnitines. You also get a lot of intermediate, that is hydroxyacyl-CoAs, and you're getting these turned into hydroxyacylcarnitines, which leave the mitochondria. Okay, now these would be your traditional hydroxy. That's intermediates, remember, in the beta oxidation pathway, right? That's right after the hydratase pathway before you get the second reduction in beta oxidation. I'm sure you remember that. So you get a lot of long chain, medium chain, short chain acylcarnitines. You get a lot of hydroxyacylcarnitines, and you get even long chain acylcarnitines, and these are presumably coming from the peroxisome, right? So that's the peroxisomal organized association that they talk about in this paper. So let's look at the peroxisome part of the model. You got, again, long chain fatty acids bigger than C16. They're being pulled out. They're being omega oxidized. Now, this is a different oxidation pathway. So you're making dicarboxylic acids, right? That's the ultimate. You're also making the omega hydroxys. The omega hydroxy fatty acids are going to be used to make acylcarnitines. Okay, they're not sure about how this occurs here. It's a question mark because that's never been described. Maybe it happens there. There must be some way of making acylcarnitines in the peroxisome because they seem to be made there. At any rate, because you can actually synthesize it and you can translocate carnitine into there. So anyways, you get a lot of C16 dicarboxylate carnitines, a lot of omega hydroxyacylcarnitines. Again, those are the ones that you end up in the blood, okay, and the heart failure patients. So they're saying that this looks like a paradigm in heart failure, right? And they're saying it's because you're getting a decrease in beta oxidation. Because why? One of the reasons is you're getting a decrease in molecular oxygen as the electron acceptor. So you're making acylcarnitines, they build up, they leave the cell, you get them in the blood. Okay. Now, what does that involve now? What is that going to cause? So let's discuss this all. Now we put everything together in what I call my concluding reasoning part. Okay. So it's not just the conclusions, because conclusions just be bullet slides. I want you to show how you go through the reasoning here. So the authors think that the corruption of beta oxidation, and they're well founded in this, I totally agree with them, is a surrogate for heart failure and therefore acyl acylcarnitines, and perhaps hydroxy and dicarboxylate, peroxisomal activity here. Acylcarnitines might be good serum biomarkers for prodromal, that is right before you get really serious heart failure, maybe right before an acute heart failure, right? They didn't distress this, but maybe. And remember, you see this in the ones that have the damaged ejection fraction and then the preserved ejection fraction. So they're not saying they can separate those two types of heart failure, but they're saying heart failure in general seems to follow this paradigmatic increase in acylcarnitines. Just saying that's good because maybe we can do a liquid biopsy. We find a lot of these acylcarnitines. We can say, aha, these people are going to either they already have heart failure or they're going to generate heart failure, and then we can go about surgery or maybe medical therapy of them. And in fact, maybe these pathway intermediates could be targets, right? Targets, because maybe these are toxic to the system, these acylcarnitines. And if they are, maybe we can target the metabolic pathways, diminish them, and in so doing, maybe pull back the etiology of the disease, right? Even with the low perfusion rates, right? Now, my take, okay, it's longer because I get to, wax here a little bit, and they put together a great discussion. I just summarized it very succinctly, right? And that's not succinate, it's succinctly. Okay, since the majority, okay, of the metabolites, the liquid chromatography mass spec metabolites, right? That's not long chain. Sorry, I put that in there. But that's liquid chromatography mass spec metabolites. Remember, they did metabolomics. Were unremarkable. And so I didn't go through all the tables. They had all kinds of tables in the paper. And they said, nothing much with ratios. There's a little bit interesting something about isoleucine there and a couple of other things, but they weren't too impressed, and so I wasn't too impressed with it. I think the acylcarnitine story is the take-home message here. So unremarkable, I'm going to call it, using like nice medical term. I love those medical terms. Unremarkable in height heart failure versus control. And there really wasn't any difference in the low ejection fraction versus the preserved, as I've mentioned. Okay, now maybe they had hoped they'd find something, but it's okay they didn't. Given all that, it's likely that the blood is not maybe such a great liquid biopsy for bioenergetic metabolome. Okay, because if you want to look at the entire metabolome, right, you might be looking for a lot of other compounds there, and they weren't very remarkable. You didn't see big differences. So maybe liquid biopsy isn't going to be great, except for the acylcarnitines. And maybe that is some slice, right, some very sharp differentiated understanding of the bioenergetic metabolome, right? So that is really what we want to look at here in heart failure, because that seems to be more critical. Right, the bioenergetics, ATP, heart contraction. Remember, that's the key there, right? All right. So the authors maybe could have, okay, this is my only criticism, and it's not really, it's just me thinking about this. Looked at serum energy charge. Remember, serum energy charge looks at all the adenolates. ATP plus half ADP divided by the three adenolates, ATP, ADP plus AMP. AMP really critical here. Sorry, that's supposed to be a bracket, not one of those there. Because AMP, high levels of AMP turn on AMP kinase, and that turns on a whole floodgate of different metabolic pathways, some of which are pathogenic, some of which aren't, some of which are actually prevent pathogenesis. So I would really like to have seen those adenolates in the blood. I know you can measure them. Maybe they got a paper coming out. Maybe they just didn't have the inclination to do it. Maybe they did and they didn't see anything. Maybe I missed it and they talked about it. I didn't see it, though. They also might have looked at oxyhemoglobin, because the amount of oxygen, that's oxygenated hemoglobin is kind of important here, because oxyhemoglobin is going to be what? It's going to be the source of the oxygen in all the peripheral tissues. So if you have enough of oxyhemoglobin, that is, of course, going to be linked directly to heart failure, right? Because heart's the one that's going to be pushing the blood around. Oxygenated blood might be decreased because of heart failure. You see how that works in terms of the cyclical argument of how heart failure can be caused by alterations in fatty acid oxidation, or indeed can result in alterations of fatty acid oxidation because of that oxygen tension. Right? I would have liked to have seen that. But it may still have been difficult to correlate to the cardiac output. Okay, so many things can result in changes in energy charge and oxyhemoglobin. And maybe that's why they didn't. Maybe they were thinking clearly and they thought, no, we're not going to do that. But I still wouldn't mind seeing it in HF patients versus controls. Now, the higher serum acylcarnitines is duly noted. That's me saying absolutely cool. However, saying, yep, that's right. I agree with them. And it may suggest a potential competitive role in fatty acid oxidation of frank cardiovascular toxicity. Now, why I'm going to say toxicity, it's going to come up in a minute. Now, when I say competitive role, if you've got way too many acylcarnitines, maybe that's going to block acyl-CoA utilization and beta oxidation. So if you got way too much acylcarnitine, way too much in that pool, not enough in the mitosolic acyl-CoA pool, bing and a bing, you don't have enough substrate for beta oxidation. You also don't have enough oxygen as the terminal electron acceptor. You get less beta oxidation. Paradigmatic for heart failure. See how that works? All right. So to put this together, this is a new feature for VerEvMed. I hope you like it. I do this all the time, but I decided I'm going to try to key it off. And maybe what I'll do is I'll make a separate YouTube video and just do postscript biochemistry. But I always want to be able to link it to the larger one. So this time I'm putting the two together on one video. So new feature for VerEvMed, exclamation point. I hope you like it. So that means let's get into some of the real intellection about what this says in terms of the population of evidence and then the verification of that evidence in the biomedical literature about heart failure associated with this particular aspect of fatty acid metabolism dysregulation. So, dietary carnitine is absorbed from the diet. I want you to understand that. I didn't mention it before. The main source or a really good source of it is actually red meat, okay, what you would expect, right, out of acylcarnitine in red meat. Less so in poultry and other kinds of products like dairy products. But anyway, you don't need dietary carnitine. You don't need to take that as an exogenous alternate medicine, homeopathic medicine, nutraceutical, because you make it. Okay, that's the important thing. So carnitine is only conditionally an essential nutrient. It's not essential. I mean, you don't have to get it from the diet. So that's a very important thing. People that go to these, health food stores and buy a lot of carnitine. I know a lot of people that try to build up muscle do that because you can imagine how carnitine might become rate limiting for muscle building, right? People that are into that. I've never seen any convincing work on that. Maybe there is. I'm not aware of it. But I don't think it's a good idea to be taking compounds that are not essential when you have plenty of it in the diet, and also you make it, you synthesize, so it's not essential. Now, caveat, preterm infants, people with a severe aciduria, now that's part of that is linked to, of course, inborn errors of metabolism. People who are septic, like septic shock because of bacterial infection, or sepsis associated with like traumatic injury, like a gunshot wound or a knife wound or something, all bets are off. Then sometimes you do need carnitine. And indeed, the later stages of heart failure, when you get major organ failure, also you don't have a lot of carnitine made because the liver is a major source of carnitine. If your liver's not doing what it's supposed to be doing, it's not going to make anything, including carnitine. Okay, so that's an important aspect. But in general, healthy people, even people that may have early stages of heart failure, probably don't need carnitine. But maybe there's a therapy there, who knows, for HF patients. Carnitine is efficiently synthesized, just to let you know, from the amino acid lysine, which is an essential amino acid. You don't synthesize that. Humans don't. And it basically trimethylysine is the precursor to carnitine. And it's made very provocatively and in very high levels in liver, kidney, and brain. So there's an enzyme called gamma-butyrobetane dioxygenase, BBD. That's the rate-limiting last step in de novo carnitine synthesis in, for example, the liver, but everywhere where it's made. And its expression is actually controlled by a transcription factor. Now, why do we want to know about this? You'll see. Remember, transcription factors control transcription, right? They go to the nucleus and they enhance chromatin remodeling and either induce or control, maybe suppress gene expression, right? So there's one called PPAR alpha. Now, that's peroxisome proliferator activator receptor alpha, okay? So there are PPAR alpha, PPAR gamma transcription factors. This is the alpha one we're talking about here. Now, what does it normally do? First of all, it's a nuclear receptor, so that is it works in the nucleus, okay? And it stimulates, its major claim to fame, it stimulates beta oxidation gene transcription. So all those genes that are in beta oxidation are turned on when PPAR alpha, when that transcription factor activates the opening up of the chromatin remodeling to cause the expression of those genes that are in beta oxidation. Okay. Now, isn't it interesting that the rate limiting step for carnitine synthesis is also controlled transcriptionally by PPAR alpha? Indeed. And it would make sense. You turn on beta oxidation, you're going to need more carnitine. Absolutely, right? So that is all, when you look at biochemical pathways, when you get down to the grainy aspects of it, it does cohere. It coheres. And you like to see coherence when you start to talk about truth, and truth is an aspect of knowledge, right? Justified true beliefs. Yeah. All right. So PPAR alpha then turns on beta oxidation gene transcription. PPAR alpha simultaneously, which is really cool, it stimulates the expression of a pyruvate dehydrogenase kinase enzyme. Now, I talked about this just recently in hepatocellular carcinoma and pancreatic cancer, okay? Ductal cancer, adenocarcinoma in pancreas. But in case you didn't watch those recently, PDH is the last enzyme in glycolysis, okay? And what does it do? PDH makes acetyl-CoA. Now, isn't it interesting that PPAR alpha will stimulate an enzyme which phosphorylates PDH, and when it does it, it shuts it down. It shuts it down so you're not getting acetate from pyruvate, which means you're not getting acetate from lactate, and you're also not getting acetate from glucose. So basically, it's limiting glucose oxidation-mediated acetyl-CoA delivery. So it's superimposing fatty acid oxidation to make acetyl-CoA over glucose. It's sparing glucose. Right? Now, this is going to link up to diabetes, right? Because remember, diabetes, especially type 2 diabetes, you get insulin resistance. Insulin resistance, or that is the insulin receptor, is insensitive to glucose. Stimulation, right? Through the GLUT4 mechanism. So that means that this couples with diabetes. And I told you, a lot of heart failure patients are comorbid with diabetes. So it's okay that you couldn't separate it out, the acylcarnitines. This is more just like mainframe. It makes sense, right? That glycolysis being turned down by PPAR alpha's activation of this enzyme kinase, which shuts down glycolytic acetyl-CoA synthesis, means that you're driving everything towards fatty acid oxidation. Okay. So starvation, high-fat diet, if you want to know what induces the activation of this transcription factor. Starvation, why is that? During starvation, what happens? Adipose tissue reserves, the depot fat is called upon to help you survive. That's how you survive. The more adipose tissue you have, given enough water, the longer you live before you die of starvation. Okay, that's the truth. All right. Otherwise, you break down protein, and if you lose lean muscle mass, lean body mass, and you can't move around, you can't gather up food anymore, so that's another way to waste away and die. So the more lipid and adipose tissue you have, and if you have a good translocation of lipid via the HDL pathway to bring fatty acid in the liver, you'll live longer. At any rate, starvation, and not paradoxically high-fat diet. High-fat diet because you have a lot of fat in the diet. The body says all this fat, we got to be able to dispose of it. So you're going to have a lot of PPAR alpha activation of those enzymes to be able to facilitate the utilization of all the fat. Right? That also happens in diabetics. In diabetics, you get a lot of lipolysis in the adipose because you're getting a lot of glucose, you're getting a lot of insulin insensitivity, so you're getting more and more fatty acids cleaved from the adipose tissue. Got a lot of circulating fatty acid. It looks like diabetes looks like you're on a high-fat diet because you get a lot of circulating fatty acid. See how that works? Not paradoxical at all. All right. And interestingly, though, if you pharmacologically induce diabetes in rats and mice, you get the same effect. But, this is like putting the cart before the horse because the pharmacological induction doesn't involve all of that interior upstream metabolic dysregulation that leads to frank type 2 diabetes. But still interesting that when you pharmacologically induce this, so we have drugs that will pharmacologically induce type 2 diabetes, that when you do that, basically the destruction of beta cells this way, when you pharmacologically do that, you get the same paradigm. PPAR alpha comes up, and that induces all this beta oxidation, which means you're going from a glucose oxidation paradigm to a fatty acid oxidation paradigm. So all has a good, coherent, paradigmatic interaction. So, more. Acylcarnitines are associated with insulin resistance. You find this in the literature. And that suggests that fat utilization superimposes as the favored bioenergetic pathway for glucose oxidation. So when you get, as I've said, you get a lot of acylcarnitines. So that's one of the biomarkers of insulin resistance. You get acylcarnitines from diabetes anyways, okay, when you get insulin resistance. Now, a reference I used for that was 2010 paper, Molecular Cellular Endocrinology, just so you know where I got some of this information. The other was 2 or 3 other sources, which were embedded in this or were embedded in the original articles you just saw referenced. Okay, so now, postscript page 2. We're almost there. Now, there is a protein called OCTN2. It is the membrane protein that translocates free carnitine, that C0 carnitine you saw build up in the blood, and acylcarnitines across the plasma membrane. So it translocates acylcarnitines out of a cell. It also allows acylcarnitines to enter a cell. Okay, that's the way these plasma membrane receptors work. These translocating receptors work. So the expression of that, wow, I was glad to find this. I found this in the literature, is actually induced by nuclear PPAR alpha transcriptional control. So you got carnitine metabolism, you got beta oxidation, and you also have acylcarnitine translocation, all upregulated by PPAR alpha, which is caused by starvation, high-fat diet, and synthetically induced diabetes. So, wow, that fits the paradigm. Very coherent, very good knowledge-based building. In fact, just as an aside, or really not an aside, but all part of the whole process, because renal failure is a big part of heart failure, also a big part of obesity and type 2 diabetes and metabolic syndrome, because you get hypertension now, for example. Renal expressed OCTN2, remember it's a translocator for the carnitines, allows reabsorption of carnitine from the urine. So that's what it normally does. Okay, so when you're filtering the water that's going to end up going into the urine, right? You're filtering back needed metabolites. One of the things you filter back via OCTN2 in the glomerular filtration process is going to be carnitine, and that's why that protein is there. Overexpression. Two more cool things. If you overexpress this protein in an otherwise wild-type mouse, which has its own constitutive amount of this, you increase carnitine levels in the heart and plasma. So it looks like overexpression of this, which could be induced by PPAR alpha 2, PPAR alpha, which as well, and what that means is beta oxidation is turned on, carnitine synthesis is turned on, right? That you get an increase of the product acylcarnitines in the heart and in the plasma. So this goes back to the heart failure story, right? All right. Likewise, if you knock out PPAR alpha, okay, knock out mice, that means you get rid of the gene, just a genomic knockout. You get very low expression of OCTN2, which is probably basal levels, and you also get low carnitine. So this all fits together. That shows that this is associated with, the OCTN2 is associated with carnitine, and the PPAR alpha is associated with OCTN2. So, cause, effect. Right. So here's the dialectic. That means now this is an opinion. This is trying to put together the information, come up with propositions, and then conclusions. That's what the dialectic is. And of course, you get this from Platonic philosophy, right? And again, I'm a real student of using the keys to categorical logic to describe the evidence, therefore verify the evidence in biomedicine. That's what I do at VerEvMed, right? I want to use logic in all of my work. I don't want to jump to conclusions, or just have opinions on this material. So let's do the dialectic together. Corruption of lipid homeostasis could mitigate insulin insensitivity, or that is another way of saying insulin resistance, via the PPAR alpha induction of acylcarnitine mediated insulin resistance. Okay, so it's the whole process here. If you corrupt lipid homeostasis by, for example, decreasing beta oxidation, you get acylcarnitine buildup. Acylcarnitine buildup is going to cause insulin resistance because you're going to limit the amount of glucose uptake. Okay, and it's going to be linked to PPAR alpha because you're going to be causing that kinase reaction on PDH which is going to limit the utilization of glucose. Buildup of glucose shuts down GLUT4, which is the translocator for glucose uptake. Voila, you get insulin resistance, right? Internally from acylcarnitine metabolism. So you do get obesity-linked. Here's another thing that we observe. Obesity-linked dyslipidemia. Okay, these are premises that are true. So the truth of a good argument is the premises have to be true. The truth of a conclusion is only as good as the premises being true, right? Because there's going to be less truth in the conclusion than there is in the premises. So the premises better be rock on hard, truthful. And truthful in the terms of scientific context means you find this in the literature. If you verify it, it looks like it's truths in terms of scientific truths. So obesity-linked dyslipidemia and type 2 diabetes are associated with heart failure, okay? So you get dyslipidemia, you do get diabetes in heart failure. So we've already described that. It's well embodied in the literature. Sorry for the pun. Heart failure could be a consequence then to dyslipidemia. This is not a surprise, but that's a decent conclusion. I'm not making the conclusion just by saying, oh, gee, well, obese people seem to have a lot of heart problems. And so, hey, yeah, it must be fatty deposits on the heart. I don't like that hand waving. I don't like any time you're going to be putting on fat anyway. This fatty deposit around the heart, sure, there's some paradigmatic evidence of that. There's oxysterol metabolism and the whole thing. But I don't like those hand-waving things. I want to see really discrete conclusions drawn. And that's what we just did here. We described here. We got some discrete conclusions from those two premises, which can be well validated as truth in the scientific literature. Now, let's continue. Besides that, okay, that's one way of looking at it. It's like going in the square of opposition, going from the universal consequential to the universal negative, okay? We can also look at subalternative understandings here. And what are subalternative understandings? We can take the same data and come up with a different way of looking at it. That's what I want you to see a dialectic allows you to do. So you don't just spin off opinions and come up, yeah, that's the conclusion, goodbye. I want you to see how you can use reasoning just with what we've read and understand it better. So here we go. Congestion or hypoperfusion because of atherosclerosis or cardiogenic shock or other cardiovascular dysfunction from any other source, okay, such as even congenital disease, right, or damage to the heart from something else, okay, is going to associate with congestion. Maybe congestion has to do with a bacterial infection that didn't go away. Maybe hypoperfusion has to do with, again, atherosclerotic plaque buildup for some other reason, like some coagulopathy associated disease, right? Something to do with blood coagulation problems, something to do with platelet aggregation, for example, which could have to do with platelet activating factor, which is another lipid. So you can come at it from, first you get that issue, okay? That could contribute to acylcarnitine buildup and then extravasation, which we know occurs in heart failure. Remember, these are going to be what? Clinical manifestations of heart failure. Extravasation. So you're going to get the acylcarnitines kicked out regardless of, okay, or maybe in cooperation with the OCTN2 protein, right, which is turned out by PPAR alpha. All right. You get extravasation, you're going to lose acylcarnitines. And again, OCTN2 mediated transport, and you get subsequent induction of peripheral insulin resistance. You're kicking all those acylcarnitines out. They're being taken up in the periphery by OCTN2, which is induced by the PPAR alpha and all in the muscle tissue, for example, or in the brain or in the heart muscle. Okay, you get all that acylcarnitine in there. Again, it's going to block glucose utilization, right? It's going to block glucose utilization because you're going to be jamming all that carbon into fatty acid metabolism. And because of that, and because of the PPAR shutting off the pyruvate dehydrogenase complex, because of the PTH kinase 4, you're not going to get glycolytic flux. Glucose is going to back channel, feedback regulate GLUT4 mobilization, and you're not going to get then the whole system of insulin-dependent glucose uptake. So it could just be that first you get the heart failure, then you get the fatty acid dysfunction, and then that exacerbates the heart failure. Right. And the reason you get, again, this buildup of acylcarnitines is because the heart failure also means less oxygen. Right? So lock it right back into hypoperfusion and hypoxia. Right? So that's what I'm talking about here. Heart failure-associated hypoxia, this is a conclusion, could be consequent contributors to decreased acylcarnitine utilization. And we know that's the case. If you don't have enough oxygen as a terminal electron acceptor in beta oxidation, you're not going to shuttle the acylcarnitine to the acyl-CoA pool in the mitosol. You're not going to get beta oxidation. You're going to, in fact, get the buildup of those hydroxys, and you're going to get all those acylcarnitines leaving, right, the mitochondria. And it's exactly what you see, right? So that's the second set of conclusions you can make. So they're not diametrically opposed, they're just subalternate understandings, right? That's the point I want to make. All right. Now, systemic coagulopathies, which you get with heart failure, can further contribute to the decrease in oxygen transport and thus inhibition of cardiac and peripheral beta oxidation. So this is all part of the whole paradigmatic system of heart failure. You do get peripheral coagulopathies. In fact, the client that I'm talking about, that was one of the clinical manifestations, was a coagulopathy. Not just in the heart, by the way. All right, so insulin resistance, final discussion here, final conclusion, could maintain the disease state. So here we're talking about, again, comorbid type 2 diabetes, right? Insulin resistance could maintain the disease state, including some instances, even heart failure itself, by decreasing glucose oxidation in the heart and skeletal muscle. So when you can't do the fatty acid oxidation because of the low oxygen tension, you'd want to switch to glucose, but you can't switch to glucose because you get insulin resistance in the heart muscle too. Insulin resistance means no glucose uptake. And indeed, there's, in case you want to know, there is an insulin-dependent glucose uptake in the heart muscle. And here's the original paper, Current Drug Targets, Immune Endocrinology, Metabolic Disorder paper from 2005. If you want to look up that citation. So that's where I got that. I had to go back and check that to see where did it first start out when we know that there's insulin-dependent glucose uptake in the heart muscle, because we already know there is in skeletal muscle, and of course in the adipose. So that ends our talk. Hopefully you got a full understanding of the paper that I was talking about. Hopefully this does do justice to our client base to show how heart failure can be understood at the level of metabolomics and indeed at the level of pathologies in lipid metabolism, acylcarnitine metabolism. Again, I want to emphasize the great work that the authors did, the Ruiz et al. Paper, and the fact that I was turned on by the podcast I heard, and that kind of helped me look at this whole thing from a different point of view. I was already looking at heart failure, but this helped me put together what became quite a different, more unique talk than it would have been otherwise. All right, so let's get to saying goodbye. Here we go. And we'll see you soon.